Hey everybody, welcome to The Long Game. We're on a mission to demystify the science that shapes our lives and to unpack how great ideas, built here in Scotland, become real-world medicines, devices, and new companies at the cutting edge of life sciences.
And Ross, because we share a passion for golf as well as science, we know it's a game of patience. So no hype, just the long game.
Yeah, I'm Ross Kagan, Regis Professor of Precision Medicine at University of Glasgow.
And I'm Sandy Weir, Tech Innovation Leader and Strategic Program Manager, joining from Edinburgh.
Hey Sandy, lovely to see you as always. This one's going to be really a lot of fun. So on The Long Game, we explore the collision of cells, code, and capital in the Scottish life sciences sector. Today, we are buzzing to welcome a true trailblazer in the Scottish biotech ecosystem, my buddy, Ishani Malhotra.
Yeah, super excited, Ross. So many of our listeners will know Ishani from her incredibly successful tenure at Carson Tech, a medtech company she founded and revolutionized 3D printed tumors for drug testing. And that work earned her the title of Woman Entrepreneur of the Year, awarded by Cancer Research Horizons in 2024. And then in 2025, she stepped down as CEO to embark on an even more ambitious journey. And Ross, you're going to tell us a little bit more about that.
Yeah, I've gotten to follow this, as Ishani well knows. She's now the founder and CEO of Oncocine, an Edinburgh-based deep tech startup. Can I say deep tech? That is reimagining cancer treatment by turning a patient's biopsy data into personalized RNA vaccines. So Sandy, let's put down our bacon rolls, and I just finished mine. And Ishani, welcome to The Long Game.
Welcome.
Lovely to be here.
Yeah, I'm so excited, Ishani. We get a chance to talk some science smack, and we get to do it in public, as opposed to normally just yakking away. So I should say to everybody, I've learned so much from Ishani. She's been really one of the models I've seen for how to do things well and right here in Scotland. So I'm really excited to have you here. You've always been at the cutting edge of patient-specific solutions. And now with Oncocine, you're tackling the holy grail of oncology, the personalized cancer vaccine. So my first question, Ishani, first of all, how did you transition from 3D printing tumors to founding a company targeting mRNA therapeutics? How'd that happen?
So I guess as my time at Casino Tech, we were actually working on quite a lot of cancer immunotherapeutics and actually speaking to various different biotechs and pharma that were coming up with all of these innovative solutions in the immunotherapeutic space. But cancer vaccines, as we know, have actually existed for a very long time. And the concept of cancer vaccines, it dates back years and years. But it's something that is one of the challenges that I recognized was that even though cancer vaccines have existed for so long, we haven't had very many successes in clinical trials. And a lot of that comes down to the patient's specific immune systems, as well as how damaged they are when someone actually is going through, having cancer. So it's about actually making it personalized, but it's also about tackling it with not just one type of data, but multimodal data sets. And I think that is something that I recognized quite early on while I was still CEO at Casino Tech, and then when I stepped down, I decided to actually come up with a solution to see if it could address those challenges.
Got it. Yes, and I had the privilege of watching this slowly be born. And actually, before we get further into it, can you do me a favor? Can you explain to our audience what exactly is a cancer vaccine, and then what is the technology you're using to make it better?
Yeah, so a cancer vaccine is essentially what we call a cancer vaccine is actually a therapeutic vaccine, which is looking at using your patient's own immune system to fight against the cancer. Now, whenever someone gets cancer, the immune system is compromised, and cancer generally has a very notorious way of masking itself so that the immune system doesn't recognize it. So essentially, what we do with the cancer vaccine is try to ensure that the patient's immune system can actually fight against the cancer. Now, this is because it's a therapeutic vaccine. It's not preventative, which a lot of people, call vaccines as preventative. This is more regarding actually ensuring that the patient's personalized immune systems can actually fight against it. With Oncocine's technology, what we've developed is an AI platform, which is an end-to-end solution that not only looks at the tumor biology and patients' unique immune signatures, and it uses that data to then discover these novel cancer vaccines with multi-target approaches, so that there is a higher chance for the patient's immune system to fight against the cancer, but also very personalized to the patient. And we can do this within 2 hours, so it's really quick.
Wow. So yeah, the 2 hours, of course, is incredible. And that really taps into, I guess, what you call true precision medicine. But I just want to jump back to a point you had made, which is, so most of these cancer vaccine clinical trials have failed, especially for solid tumors. Yeah. So right now in the UK, we do have, I could find a couple of them, the BNT116 lung cancer vaccine, the BioNTech colorectal cancer vaccines. So these are ongoing, and hopefully they'll work. But the track record, as you said, is not amazing. So why? So you take, as you say, you take a much more comprehensive approach. You're accounting for mutations, infiltrating lymphocytes, all this stuff, the spatial, the tumor microenvironment. So why does that matter? Why is that a better approach, and why is that going to succeed?
Yeah, so that's a good question actually, because so historically a lot of the cancer vaccines have been developed using binding only predictions, or essentially just using the genomics or mutation data from the patient. But as cancer biology in a patient is always evolving and always changing, and new mutations pop up even before we realize, and that's why there's metastasis and things that go along that. So it's very important to actually have something that is adaptive and in real time, but also actually looking at other aspects of the patient cancer biology, because the tumor microenvironment, and there's tons of publications and peer-reviewed literature out there to talk about how tumor microenvironment is crucial, and spatial transcriptomics is crucial to actually look at various different tactics for immunotherapeutics, and how it can actually be more targeted and better approach if you used tumor microenvironment and spatial transcriptomics data. So this is something that it's not been done in vaccine design before, but this is something that we thought would be a very accurate approach of actually looking at the patient's specific tumor microenvironment and spatial transcriptomics, alongside the mutation and RNA-seq data that you would generally get from the patient. But alongside that, we're also looking at combining it with imaging data, and in the future, even looking at large imaging data, and so that we can actually get a full body approach for the patient as well. Because ultimately, you want the immune system to fight against the cancer. You need to understand exactly what's going on in the body, not just in from the mutation side, but overall. And that's why this approach is something that would give a lot more accurate results. And in fact, we've just recently tested with one of the BioNTech clinical trials that have been ongoing. And I can say very, happily and confidently that we've got some fantastic results, much, much faster than ongoing clinical trials at the moment. And that's something that has been really crucial to actually build that confidence in our platform as well.
I have so many questions. Oh my God. I love this. And so a little bit for the nerds, we're going to have to go a little bit in the weeds. So I apologize to everybody else, but just I want to dig a little bit deeper, because you said some of the things that I love, which is taking a whole body approach. As you know, this is what I've always been about. And I know for you as well, actually, the technology you've had has always thought about that. But so let's take one quick dive in, which is, So typically the way you make a cancer vaccine is you say, oh, okay, I sequence the tumor. I see these weird proteins being made. I now try to train the body to recognize these weird proteins. They're only on the cancer, and then go let your immune system go find them and kill them. You mentioned that you also take account of the tumor microenvironment, all the stuff around the tumor. And for those of you who aren't cancer people, this is a big, big contribution to the tumor. And in some ways, it's what makes getting tumors down so frustrating, because we know so much about the tumor itself, but then you have to account for all the other stuff around it that helps it progress. So that's just me ranting to say, how in the world do you account for the microenvironment when you're designing these vaccines? Like, what are you looking at?
Yeah, so that's a good question. So what we look at with the tumor microenvironment side of it is actually, so the tumor microenvironment is a very complex, and as Ross mentioned, very complex system where you've got various different cell types that are at play there. It's not just, what the cancer does is essentially surrounds itself with all of these cell types that is almost protecting it, at various different points with various different pathways and everything that's going on there. What we're trying to do is essentially not just look at the cancer cells, but also look at these various different other cell types. For example, if there is a tumor microenvironment that has a high stromal or what we call fibroblasts or CAFs, that's cancer-associated fibroblasts within the microenvironment, it essentially forms almost a shield. And actually, there's been a lot of papers actually to show this, where immunotherapeutics find it very difficult to penetrate. Because of the shield that it forms. So if we are able to account for that in when we are developing and designing these vaccines, for example, if a patient has a high infiltration or high stromal index, then we look at that as part of our vaccine design. And essentially, the platform then not only gives monotherapy suggestions, because a lot of these tumors would be considered what we would call cold tumors, and that would mean that an immune response or the patient would have a very immunosuppressive environment, and so for that reason, we would not a monotherapy vaccine, for example, would not necessarily work, and that way the platform recognizes that. And gives those suggestions as combination therapies with existing checkpoint inhibitors, for example. And this is something that you can probably, take the example of the Moderna Merck trials that were ongoing, where they had a combination of a checkpoint inhibitor with the vaccine, and there was a 44% uplift in the response. So definitely it's something that, but we are able to recognize that before it even goes into clinical trials, which means it can give you those suggestions right, when we're discovering the vaccines, and it's part of the vaccine design.
Got it. Got it. So let's okay, I'm going to push this a little bit further. And that is, so for example, when I ran an experimental clinical trial back when I was in New York, and I always think about one patient in particular where we designed a bespoke treatment for them. We designed a model based on their sequencing. We treated them. The tumor went down. But then at some point, it came roaring back. We all know this phenomenon. And when we resequenced it, we saw that a small subclone, when we looked back, was about 5% of the original clone, contained EGF receptor, and then that thing just ignored what we had just created that didn't target that, came roaring back because now we had opened the niche up, and it was a problem. So you, by being able to redesign these things in a couple of hours, now of course that's not making it, but that's designing it. What is the prospect to be able to walk these tumors down by being really sort of back and forth interactive? Let's put the cost aside for a moment. Obviously, at the beginning, that would be pretty pricey, but let's just dream a little bit. Given what you're opening doors to, what would the prospects be to walk these tumors down and adapt as the tumor adapts?
Yeah, so that's actually a great question, because our platform, we've recently added in an adaptive vaccine discovery side of it, which means that what we would be looking for is essentially a patient comes in and they give, their biopsy and blood sample, and we would use that data, that, whether irrespective of where they are in the world, and, whichever hospital they are going to for the biopsy and the blood, we get the data, we put that into our platform, and then 2 or 3 months later, when the patient has a follow-up, they give blood again, and then we use that data again. So essentially, we follow the journey of so it's more longitudinal. We follow the journey of the patient, and ultimately the platform itself starts to learn and pick up on these immune signatures that are changing, what we like to call certain immune signatures, certain immune health of the patient that's actually changing over time, and that then ultimately starts to pick up those markers, whether it's biomarkers, whether it's neoantigens, whether it's any of these changing mutations. It starts to pick those up, and it starts to learn, and then when the platform learns, it then starts to predict ultimately what the evolution basically of the tumor within that patient. And that's when we start to suggest various different doses, because that means that maybe a patient requires, one dose of a particular vaccine that we've designed, but then maybe 3 months later, there's been a drastic change in the patient's tumor biology. We then design a second dose for that particular patient's, which is very personalized to the patient again. And that's what we call the adaptive vaccine discovery, which the platform is now starting to do and pick up on. And of course, the one of the biggest things is we want to test this with some of the cancer institutes we want to collaborate with, and essentially see whether it's actually picking up a lot of those changing mutations quickly enough, and able to design that quickly enough. Because as our platform does it within 2 hours, it's actually saves a lot of time, and we also get manufacturing ready outputs, which means that there's actually a lot of saving in the manufacturing side of it, because if a manufacturer gets detailed instructions of how to manufacture a vaccine, then that saves a lot of time and money as well for not only for the ultimately for the pharma, but also for the patients.
I see. So part of the answer to the question of sort of evolving with it is trying to look prospectively and trying to predict where it's going to go. And in other words, your approach is not the rate limiting step. It'll be the manufacturing, and you guys are actually thinking about that. So that was part of also my question is, yeah, thinking about it. So that makes sense to me. So in fact, I guess you could say you're kind of taking a network approach, using AI to think about the problem. This is why I totally love this, I should say, because I'm on a soapbox at least once a day on this with my lab, which is you're really trying to think of it in terms of a larger network problem and not a I see tumor, see tumor, shoot tumor, but trying to see it as a larger problem, both spatially in the whole body and also temporally. Things change, all that stuff. And this, you're really leaning into the use of AI, which takes me to a question I've always wanted to ask you. Okay, I'm sure you're curious what on that lead up, which is you and I have talked about your need for really high level coding and so on. I have seen what you have personally coded. I'm pretty sure you're not a hardcore coding jockey, but I've seen your Oncocine work. Okay, it's pretty impressive. This feels very 2026 to me. So tell me, how in the world are you doing this and getting these tools done with creating this OnVac that you're doing? Basically, how do you do this, and how far can you really push things?
Yeah, so it's one of the things where I think I learned to code when I was doing my biotechnology in my bioengineering days a while ago, and coding to develop microfluidic chips and things like that, which was really interesting. But I was very used to working with Python and BioPython actually, and I continue to do that even now because I find it I understand it a little bit better. But of course, there's actually quite a quite good tools out there now that can help
Mm.
you, account for errors and things like that, which is something, can be quite frustrating if you're coming up with a massive platform and code, where it could be one small parenthesis that is out of place, and the whole platform it doesn't work. So I think that one of the things that I guess I started, my approach was more about looking at Bright with my knowledge and with my expertise and some of the papers that I was reading continuously about, the tumor microenvironment and how it influences combination therapies and things around immunotherapeutics, and how it can actually be very helpful in understanding personalized immunotherapeutics.
Mm.
Um, it gave me a bit more of, okay, if I can come up with some algorithms that could be really interesting to include in the vaccine designs, then that could be interesting. So our algorithms are all proprietary, because it's something that I've come up with that sits within the platform,
Yeah,
and
noted.
then the coding around that is just uh, coding that we've got, but then it's all very specific to the algorithm. So the whole platform is built from scratch, but a lot of the errors were fixed with using some of the existing coding tools like Cursor and even Claude Code, for example. But yeah,
Mm.
like I would say that because the platform itself is something that has been essentially an amalgamation of not just what my experience has been, but also from some of our team members that have come in together to actually tell us about their experiences and how we can shape the platform to better reflect what would be the need for pharma, biotechs, and patients ultimately. And that's something that allows for the algorithms to keep changing as well as adapting. So I think that keeping it very specific to what the market needs are is also really important, and that's something that we can do real time as well. But
Mm.
yeah, the actual platform itself, it took me about 6.5 to 7 months to actually build it, and when I was happy with
Wow.
it, and then we started to do a kind of like first trials with published data sets, and comparing to peer reviewed publications, and we started to see that it was giving us some really fascinating and really good results. So yeah, so I think it's at this point now where we are really happy with it, and we are we're continuously testing now with various different patient data sets.
So this is so amazing. So because the reason I'm saying this is amazing is not just you're a guest and I want to be nice to you because I really do like you, Ishani, but it's because, they often talk about now there's going to be the first $1 billion single person company and all of this stuff, which hopefully you'll be that person and then you can invite me to your island. Um, but this is really an example where you've leaned into the tools that are coming online and using your knowledge. You've actually done quite a bit of this yourself, I think is what my point was and why I'm so
Mm,
impressed
mm,
by this. And you'll be too polite to say that, but actually what you've been able to do as a single entity, and I'm sure you've gotten help, I'm really impressed.
Mm.
Obviously, all of this requires, serious computational power and all that stuff. So now would be a perfect time for me to hand this over to Sandy to talk about and
Thank
think
you,
about the
Ross.
tech and business side. What do you think, Sandy?
Yeah, that's a great segue. Thank you, Ross. Yeah, Shani, this is so impressive. I've really learned a lot just listening, and as you can tell, so Ross handles the biology, and now I'd like to talk a little bit more about some of the kind of practical adoption of some of the technologies you've been describing. I've just returned from a really stimulating week when I've been at Precision Medicine World Congress in Santa Clara in California. By the way, what you're talking about and your world and your company, you would fit in so well to those discussions.
mm,
I have heard a huge amount this week from companies like yours using AI-driven multi-omics models, and you're absolutely so right. It's that stack now. It's a yeah multimodal stack, and using that to unravel complex tumor mutations and understand immune responses. I think what you're doing is fantastic.
Thank you.
Let me come back to that question on speed, and you've talked very eloquently about the Oncocine it's called the OnVac platform,
Yeah,
isn't it?
yeah.
And you take a patient sample and you generate that fully optimized, manufacturing ready microRNA vaccine, and you do that in just 2 hours, as you've told us. It's so impressive. But from a tech perspective, just take us back a little bit and tell us a little bit about how you build that automated precision pipeline that's capable of moving that fast to support it.
Yeah, so I guess one of the things that we've been looking at is actually there is quite a lot of published data sets that we can work with and use currently, and it's something that it's getting better, it's growing, but then there's also the aspect of actually working with some cancer institutes, as I mentioned, to try to get some data from that could help with the initial data set side of it. But ultimately, we do want to get to a point where we have our own proprietary data sets as well in house, and that's something that is going to be crucial for us down the line when we start to look at patient stratification strategies and things like that in the near future. But the actual technology itself, because it's an AI platform end to end, it has various different data classes that it's made up of, and it essentially ultimately goes from taking that data, processing it, and then finding those or discovering those novel neoantigens, some cryptic peptides, and then also ultimately going to looking at the manufacturing side. So because we are looking at mRNA specifically, mRNA, with COVID vaccines and stuff, we saw that mRNA has a faster manufacturing timeline. They are not as toxic as the rest of the other, peptide-based vaccines
Yeah.
side of it, and ultimately they are. They can be optimized to have to be a bit more personalized as well. So that's something that we picked up on the manufacturing side, where we looked at, okay, if this mRNA had to go into manufacturing, what would be the parameters that we would need in order to ensure. So there's like a few algorithms there that cover that as well. So ultimately, you get a kind of a very detailed report alongside the vaccine discovery and alongside the manufacturing report, which could then go straight into manufacturing. And we're looking to collaborate with a couple of manufacturers in the mRNA space to help us with some of that real world manufacturing side as well.
Yeah, no, that's great to know, great to understand, and I mean, and if I understand, you're offering, from what I've gleaned and what you've told us today, that speed is a massive commercial advantage. So that ability to be able to develop these vaccines in 2 hours, that's your USP amongst all the brilliance of bringing together this ecosystem and creating that capability. This time that you're able to the time that you're able to develop these is the leading part. And I know that you're doing things like you're participating in the Advanced Therapies Integrates North 2026 event that's coming up, and I know that you're really involved in networking with other industry partners, other academic organizations as well, including PrimeMed, which I believe you're now becoming active in. I'm really interested to know that how do you use those cross industry collaborations, and how do they help you to scale deep tech AI platform development in Scotland?
Yeah, so that's a great question. So I do always, and I've always believed in this, that collaboration is key when it comes to to working with especially when you're doing something that's deep tech, and when you're looking at something very specific in terms of coming from the cancer biology side. It's so important to see what what research is going on within academic institutes, within industry, to see how better we can collaborate with what's already existing out there, and what's already and for us to have a shared mission, to actually ultimately get to a point where we are, helping patients. So I think that collaboration is key. It's very, I think it's with Scotland's ecosystem, it's always been great for, small companies that are starting up. But I think now there's a lot more focus on actually getting cancer institutes involved and academic institutes involved as well, which is something that wasn't, when, for
Mm.
example, when it started Cocinotech, it wasn't as prevalent as it is now. And I think that's really wonderful because there's just so much amazing research, that's happening in some of these amazing institutes that we've got here in Scotland, that we need to utilize that as even industry partners.
Mm.
So I think that's really crucial as well. I guess even looking at promoting that, entrepreneurship side, I think it's really crucial because there will be a lot of there's a lot of interesting innovations that are coming out of these academic institutes, but maybe they
Yeah.
don't have, the right kind of platform to spin out or ultimately get to a commercialization stage. So that's something that, with these collaborations can really help with, for example, Primed, as we just spoke about. So that's
Yeah.
ultimately set up to help a lot of scientists and postdocs and PhD students to actually think about entrepreneurship as an option as well.
Yeah, that's great to understand, and I'm so glad that that community that we have in Scotland is really, really working for you and helping to promote you and your company. I find that incredibly positive, and certainly that's the type of interaction that Ross and I are really keen to both support and promote. So lovely to hear that reflected back.
Yeah, I think,
So,
sorry, I just want to
so
say very
please come in,
quickly.
Ross. Yeah, yeah.
Yeah, sorry, I just want to say very quickly, and it does help that, Ishani, you've been very aggressive about connecting with various people. I've
Mm,
Mhm.
seen you move through, through P4. We can
mm,
certainly talk about that as an accelerator fund. You've met a lot of companies there. Also, just moving around Scotland, it seems like everybody I know who's in that world knows you.
mm.
So I think some of it is you've really given sort of a lesson on how it is to connect to people and then, reach back when you need help from them. And you've certainly given a lot of help to others that I've seen as well.
Mhm.
Yeah, it's so important. So important to be prepared to participate in that way. Really put yourself out there, take the risk. What's the worst that can happen? And of course, you're reaping the benefits now, Shani. And you've proven you've been so successful already. I mean, you've proven what you can achieve already with Carcinotech, and with Oncocine. I'm really interested now to understand sort of if you look at your next 12 to 18 months, are you able to give us a little bit of direction around what your strategic roadmap looks like and where you're going?
Yeah, so I guess with Oncocine now we've started to onboard our team as well. So we've got a CSO and a fractional CTO that's joined us, and we're now looking at I guess the first few months have all been about validation, but we're
Mm,
going to continuously validate with some of the peer-reviewed and published data sets that we have got access to, and then ultimately collaborating with some cancer institutes to access some more data sets and also do some wet lab validation as well. So that's something ultimately we are looking to do, and I guess in the next 12 months or so we are fundraising as well. So we've just kicked off our fundraising round. Yeah, so we are looking to get some funds in to actually do some of that wet lab validation, but also do some early pilots with some biotechs and pharma that we're looking to speak to as well. So ultimately, we want to be able to get some market feedback as well. We want to be able to get some commercial, and we know, and this is something that I learned with Casinotech as well, that your offering changes based on the market feedback, and that's something that's really crucial for us in the next 12 months. So working with some cancer institutes, pharma, biotechs will give us some good feedback that will allow us to then shape the offering, and offering for the better, actually. So yeah.
yeah, no, that's great to understand. And you kind of answered what my follow-up question was going to be around, where do you see your primary partnerships, and I think it's really the sort of pharmaceutical side that you're looking to partnership with most strongly. Just tell us, maybe just the sort of final question for me is, just help me understand how you're managing that clinical translation. Are you working with clinical partners in Scotland? Are you looking to work more internationally? Is it a bit of a mix? Give us a little bit of a feeling for how you are going to do that clinical translation, please.
Yeah, so it's definitely a mix of both. So we're speaking to a few cancer institutes here in the UK and Scotland as well, with Ross's group as well. But we're also looking at a couple of hospitals or cancer centers we're speaking to in the US. And ultimately, I mean, we're not, as long as we've got some few strong collaborations where we can look at some, wet lab validation, but also help, have a co-development partner that can help us with some of the early trial data that we would want to, have in place for some of these cancer vaccines that we're already discovering, and from the platform. So it's something ultimately we need to be able to see in vitro and in vivo how these vaccines are working. And I mean, we've already got some very good data to show that our platform is replicating some of that in vivo and even clinical trials data. But we ultimately need to have some of it for our novel vaccines as well. So that's what we are looking at. And it's we're not just looking at the UK. We are actually looking at Europe and US as well for those collaborations.
Oh, that's great. Got it in one. Yeah. And listen, Ishani, it's incredibly exciting watching how you're developing and how your company is really pushing these boundaries and AI and multiomics. I hope that you're able to tap in really strongly to the kind of ecosystem that Ross and I are really pushing and supporting through Scotland. As you've said, this is a multimodal game, and there are lots of different specialities and capabilities in Scotland in terms of the data that we're pulling together and how we're applying solutions to that data. But taking this to the global market, absolutely fantastic, really inspiring, and it's great to see a Scottish company doing this. And thank you so much for joining us today. I've learned a huge amount in just a very short time.
Absolutely. Ishani, Oncocine is exactly the kind of innovation that will basically bludgeon cancers into manageable, curable conditions. And I will hold you to that. So we can't wait to follow it up.
Thank you.
Indeed. And Ross, we're coming to the end of our episode today, and we love talking about the science and the business side, but we like to bookend every one of our episodes with something that you've christened a wee bit of joy. So before we go and before we skip out of this episode today, it's been an absolute pleasure talking about the algorithms and how we get those from labs to benches to clinical practice. But for our closing segment, let's think about a wee bit of joy. So Ross, can you kick us off? Can you give the direction to Ishani here and help her out? And what's been enthusing and exciting, and what have you been getting joy from this week?
Well, let's start with Ishani. Let's start with you because, of course, this podcast is a wee bit of joy. But besides that, I'll share my wee bit of joy for sure. But actually, you're the guest. Tell us, what's been making you happy?
Does it have to be just this week?
Whatever you want to share with us. 100%.
Yeah,
whatever you want to share with us.
Yeah, I guess one of the things, I absolutely love driving around Scotland. I think it's beautiful and magical. So yeah, I think a wee bit of joy is definitely, some of the road trips that we've done around Scotland. It's been really, really beautiful, and I love driving on challenging roads, which has been really interesting. And it's not, I have a very old car, but it works really, really well on these interesting terrains. So yeah, I ultimately love that. This week I would probably say that I guess I actually mentioned it in the podcast. Was one of the biggest things or biggest joys that I got was actually looking at some of the data that we were comparing with the BioNTech clinical trials, and it was really fascinating. When you start to look at these amazing results, you're just like, okay, this is you're onto something great here. So yeah, I think that was ultimately really interesting. Spoken like a scientist, but yeah.
I was going to say that is the nerdiest possible answer. I love it.
I know.
And Ishani, I was trying to help you out. I was hoping Ross was going to lead the way, but Ross is quite right. He let you go first as our guest today.
So now it's so Sandy, you got to get. I love that nerdy, and also with your old car, Ishani, I look forward to finding you somewhere in Scotland and picking you up.
Yeah,
North Coast 500.
Bailing you out. What do you think, Sandy? What do you got?
Well, for me this week, it was more of a huge piece of ecstasy than just a wee bit of joy this week. Ross, as you know, I'm a huge Scotland rugby fan, and it would be remiss of me not to mention Scotland's resounding victory over what is a hugely respected France team on Saturday. I guess it was great to see the result, but I was particularly proud of Gregor Townsend. He's been a manager, a coach that's been under huge pressure recently, and to see him cope with that, and to see the team cope with that after a difficult start to their campaign against Italy in the opening game, it's been fantastic. And I guess we've got our fingers crossed, and we're hoping that on Saturday they can do something really special against Ireland and take that historic next step. So yeah, that was my bit of joy this week.
I love it. I love it. I actually know, I understand a little bit about it because my wife Yumi and I have been watching the Six Nations on Netflix. So I understand how terrible it was to lose to Italy and how amazing it is to win against France. So so it sounds like a roller coaster. I do appreciate it. Yeah. So my wee bit of joy is just a quick story. So on Saturday, Yumi and I went for a lovely hike at the Wangi, and as New Yorkers, I'm always blown away by actual mountains and that sort of thing. So it's always and sheep still blow my mind. You guys let them outside; it's amazing. But that night we got tickets actually because Yumi knows somebody who knows somebody, because it was completely sold out, to see David Byrne at the local Armadillo. And for those of you who don't know David Byrne, he was head of the Talking Heads, which was a huge band. For a New Yorker, really means something back in the late 70s into the 80s. The guy's just brilliant, and he hasn't gotten any less brilliant, I have to say. He comes out of the Rhode Island design, and so you could see all of that in his stage sets and everything. It's just incredible. I should probably show pictures. Maybe we'll put some up on the website. So here's where I'm going with that. So we before we saw the concert, we were eating at a restaurant mitzvah, and there were 6 people on our left. They were about our age. They were yakking. So we started to yak with them. Turns out they're going to the concert, right? The people on our right, also about our age, they were going to the concert. Okay, so it was so interesting. So we go there. The concert. And I have to say, it wasn't just people our age. In fact, we were definitely the oldest people in our section, probably because we were in the nosebleed seats, and all the people our age were down there because they could afford better tickets. Ours were free. So anyway, but as he was going through it, and I was looking at Yumi, and it just kind of threw me back to when we first met, right? Because that was around that era. And I looked around and I saw other people our age, and they were having that same moment. You could just see it. Oh, yeah. And I'm not somebody who really thinks much about the past. It's probably not a great thing of mine. For me, if a song is more than 6 months old, I'm like, eh. But I realized that just the beauty and the power of having these touchstones in a relationship that has gone on this long. And, Yumi and I were just having that moment. And I have to say, I absolutely love that. And we're still kind of on a little bit of a high, just kind of walking back into that moment. And I'm sure others from that concert had the same. So for those of you who are younger and just getting together, just look forward to those moments. It's kind of what makes life absolutely special. So that's my wee bit of joy.
That's beautiful, Ross. And yeah, I'm a huge fan of David Byrne as well. So
Yeah,
I'm so jealous that you had this time at the concert. Yeah,
it was
fantastic.
amazing.
That's a lovely story.
Thank you. Well, guys, next week we will talk about the fundamentals of cancer. But before we go, we have a thank you to Ishani. Ishani,
Absolutely.
much appreciated. It's a ton of fun. I hope you had as much fun as we did.
Yeah, I did.
Thank,
Yeah, thank you so much for having me. Yeah, that was really good.
yeah,
Yeah, and,
thank you so much for your time today, Shani. It was a really tour de force of your background, your company, and very inspirational. Thank you very much.
Thank
yeah,
you.
much appreciated. Now get out there and cure the cancer.
Yeah, exactly.
So, so until next time, everybody, please have an epic week playing the long game. Ciao.
Bye for now.
Yeah, see the vision clear. We're here to run the long game. Blueprint set, no fear. Playing out the long game. The long game.